Okay, well, welcome everybody to workshop number 31, Fundamental Discoveries to Elucidate CFTR Cell Biology and Ion Transport. We have a really interesting lineup of talks today, and I'll just give you a brief overview of the speakers. So first off, we're going to have John Hunt. He's going to talk about cryo-EM studies cryo-EM studies of human CFTR folding and conformational dynamics elucidate a potential, a novel potentiator binding site in the nucleotide binding domains that supports an alternative channel gating mechanism. That's going to use up a lot of the time. We have a 15-minute session for each speaker. There'll be a warning light here for the last 3 minutes. And then we'll have 5 minutes of discussion of each paper. And cryo-EM studies have provided important insights into CFTR gating and modulator binding sites, but there's much that remains to be learned regarding the mechanisms of CFTR channel gating. So John will present new information on protein folding and its implications for the development of new CFTR modulators. The second speaker, Jean-Pierre, CFTR is expressed in many cell types, including endothelial cells that are highly mechanosensitive and implicated in pulmonary edema. CF impacts the synthesis and distribution of lipids that determine membrane fluidity and modulate mechanosensitivity. So Jean-Pierre is going to present recent studies of the relationship between CFTR and the mechanosensitive channel piezo-1. The third speaker, Carrie Thrasher from the University of Alabama at Birmingham, is going to talk about CFTR modulator therapies that are unlikely to help people who do not synthesize full-length CFTR, but a small molecule that increases the read-through of stop codons has been identified that acts on the release factor ERF1 that normally binds mRNA and terminates translation. So Carrie is going to discuss how ERF1 is targeted for degradation and the mechanism of action of this potential therapeutic. The fourth speaker, Christina Barilla, is going to talk about well-differentiated primary cells. They remain the gold standard for assessment of CFTR modulators and precision medicine in vitro, but are difficult to standardize and problematic for genome editing and scale-up. So Christine will talk about the differentiation of airway epithelial cells induced, prepared from induced pluripotent stem cells, iPSCs, which offer a potential solution to these problems. And our final speaker, Viral Shah, is going to talk about the label-free identification of cell types in airway epithelium. Airway epithelium is a complex tissue with, as we've heard at this meeting, many different cell types. Isolating the various cell types for studies of their dynamics and functions is a major challenge. So Viral will talk about label-free 2-photon imaging methods that will use autofluorescence and machine learning algorithms to identify the cells and also novel structures in the airway epithelium. So with that, I'll ask the first speaker to come up. John, if you could. I'm just going to load your presentation here. And you're going to work the timer, are you? Yeah. Okay, good. Okay.
Is it possible to get the cursor on the?
Yes.
Use the pointer here.
Yeah, perfect. Thank you. I'd like to thank the organizers for the second opportunity to present at the meeting. Yesterday I talked mostly about folding, and today I'm going to talk mostly about channel gating and potentiation. So the people involved in the work, the cryo-EM was done largely by Blaine Laughlin, Chi Wong, a little bit by Sergei Voroviev. Georgy Lukash has been a key collaborator for the last couple of years. Andrei Alexandrov from Jack Reardon's lab did the electrophysiology I'm going to show you. And then this is a team of people I've worked with for about 2 decades with sponsorship from the Cystic Fibrosis Foundation. John Kappas and Wendy Yang and Christy Brett from UAB. We initially started working together on for studies of MBD1 folding, and then for the last 7 years or so, we've been doing folding CFTR. Ina Urbash from Texas Tech, and he was also involved in thermodynamically engineering the CFTR constructs that we use for our studies, or most of them. And Joachim Frank is my collaborator at Columbia, who taught my group how to do cryo-EM. So, and Ollie Clark is another Columbia faculty member who helped out with some of the more technical aspects of this. Okay, so my lab has been focusing on well, since its inception 20 years ago or so, on trying to elucidate complicated molecular mechanisms. And the advent of cryo-EM is just like a revolutionary advance that enables us to see all sorts of amazing details of protein structure and function. And one of the things we can now begin to see is that there are many, many different conformational states that proteins adopt, and that these are likely to be key features, functional features, and that the static view of protein structure as being, sort of this conformation or maybe on or off is just really way too simple. And I think electrophysiologists always knew this because they would see all sorts of detailed substates, and now we're beginning to have enough resolution in structural studies to see more of that. So CFTR, so my lab has been focusing when we're doing cryo-EM particularly and try to identify every conformation that's present in the sample. This is different from what most groups are doing. Some other groups are doing it in the same goal that we have. We're all doing it different ways technically. It's a big issue now in cryo-EM methods development to try to find a systematic way to do it. But we call it global conformational ensemble reconstruction, which is a systematic attempt to find everything that's there. And so in most CFTR samples that we look at, we can find actually most of these states. Some samples show all these states simultaneously, but all of them show at least 3 or 4 different distinguishable states that are reconstructed at near atomic resolution. So the basic gating cycle, the big level picture would be the totally inactivated state of CFTR has the 2 nucleotide binding domains pulled apart. ATP binding at their interface pulls them together. The 508 del mutation, I'm not going to talk about it today, and I didn't talk about it yesterday explicitly. We have a lot of cryo-EM data on 508 del. It's a bit more complicated, which I'm not showing that today. But one of the main things it does is it induces dissociation of the nucleotide binding domain, which then unfolds and is targeted for proteasomal degradation. But what I'm going to focus on today are, and we suspect, I didn't say this yesterday, but I think there's good reason to believe that this domain-dissociated, MBD1 dissociated state is actually the source of the flickering conductance states that are seen in some electrophysiology experiments where you have an unstable channel that's opening and closing. Because the nucleotide binding domains are not holding it together tightly. So these states all have the MBDs associated, but they differ. There are 2 different conformational changes that are interrelated, and this is the main thing I'm going to tell you about today. The nucleotide binding domains flip between the canonical pseudosymmetrical state that is characteristic of the ATP-bound state of most ABC proteins, and then a distinctly asymmetrical state. There's also a second conformational change that takes place in the exit portal region, and the relative population of the exit portal conformations depends on the conformation of the nucleotide binding domains. That's what I'm going to tell you about today. And in the process of studying that, we've found what could be a novel potentiator site. So the degree of inter-NBD asymmetry modulates the conformational change in the exit portal, which I'll show you the structure in a bigger view in a moment, but basically 75 angstroms away. So it's a long-range allosteric conformational change. Several lines of evidence suggest the non-canonical asymmetrical inter-NBD conformation may be the most active state. So I'll show you both lines of evidence this morning. MANT ATP, which is a benzoic acid derivative of ATP on the ribose of ATP, potentiates the channel and favors the non-canonical asymmetrical inter-NBD configuration. I'll show you in detail why that's true. And active G551D CFTR has asymmetrical NBDs, and it's very unlikely to ever adopt the canonical symmetrical conformation. So for those 2 reasons, we think it's likely that the asymmetrical non-canonical state is probably the most functional state of the protein. So if you look at the baseline structure has symmetrical NBDs and what looks to us like an included exit portal, although that's a tentative conclusion or hypothesis. In any event, if we look at the sort of interior of the channel in the NBD-associated state, I should say the channel doesn't exist in the NBD-dissociated state. There's just nothing there in the middle of it. So when the NBDs come together, it forms this sort of fat internal channel. But down in the extracellular region, there's a very tight constriction that is definitely smaller than a chloride, and it's like much smaller than an iodide. So that has raised questions about what is really the open state of the channel. I'm not going to resolve that today, but obviously I have thoughts I'd be eager to talk to you about later. Our thought is that these fine conformational transitions, and especially because there's allosteric communication between the NBDs and the exit portal region, that they may be actually modulating the state of the exit portal region and opening it up more widely. I'll show you some evidence that's true. We definitely don't know that that's definitively the case. So this is just a quick technical summary. And again, what we're doing is we're doing an exhausted, but it's admittedly somewhat subjective search for all alternative conformations in a sample. Once we've seen once we've gotten a structure at reasonable resolution, we can find very small amounts of it. But until we've seen it, it can be hard to find it. And there are definitely, probably still some hidden conformations that are present at some level in most samples. And anyway, so this is, I'm going to explain now how we sort of schematize the color our CFTR structures. These are electron density maps, I should say. So I'm going to present NBD1 is colored blue, ATP is yellow. This is a little bit orange-yellow because I think that's actually MANT ATP. MANT ATP is going to be shown in orange, ATP in yellow. The NBD2 in green. The first structural transmembrane domain contains 4 helices from the first sequence membrane spanning region, and then 2 helices from the second one, and so that's domain swapped. So it's 4 plus 2, or 6 total, but coming from different sequence regions. And the second sequence region is shown in light green. The first sequence region for the transmembrane domain is shown in light blue, and the lasso, which is an N-terminal extension that's critical for folding, as I explained yesterday, is shown in cyan. And the F508del mutation, for reference, occurs at the interface between NBD1 and NBD2. The G551 mutation is directly in the ATP binding site that's right in front of you here. And just like with knowing almost nothing about the structure, you could know that this is going to be a serious problem for ATP activation because it puts a negatively charged residue immediately adjacent to the gamma phosphates of ATP, and it's clearly going to be an electrostatic repulsor problem. And the structures I'll show you will show that that's the case. Okay, so yes, and the extracellular exit portal is down around the back here. So this is an example of a global conformational ensemble reconstruction. These are 4 different samples that all gave 6 different structures. 4 of these are the symmetrical and asymmetrical version of the NBDs with the different states of the exit portal, and then we have an NBD1 dissociated state in a completely closed channel conformation. So what's shown here is one sample that is, just, and this is our standard construct, and I should say it has a hyperstabilizing mutations in NBD1, and it has a hydrolysis deficient mutation in NBD2 that tends to lock it in the ATP-bound state, but in digitone in the channel does not form the channel as easily as it does in membranes, and so it's not a perfect lock in digitonin. So the orange, so these are populations. This is the fraction of the population, these different samples. In addition to VX770 does absolutely nothing, and we've done this repeatedly in different ways. In digitonin, VX770 has no effect. It binds to its nice little site. It does not change the conformational distribution at all. But MANT ATP produces a significant shift, and in particular, the symmetrical states of the NBDs go down, and the asymmetrical states of the NBDs go up. And there is somewhat of a greater, actually, closure of the NBDs, and we think that's because the MANT is adding extra binding energy that's pulling the NBDs together. So this is electrophysiology data from Andrei Alexandrov. So what we've done here is he's compared F508, this is wild type, this is actually wild type CFTR, and this is with a 0.2, actually this is with saturating ATP 5 millimolar. The PO is about 0.28 at 25 degrees. In the presence of MANT ATP, now reduced, this is actually, because of the way we do the cryo-EM, he first did the experiment with 0.2 millimolar ATP and 0.5 millimolar MANT ATP, and the PO increases 2.5-fold. He later went back and did it with entirely MANT ATP, and it's exactly essentially the same PO. This is showing F508del, or a hyperstabilized version of it, and MANT has the same stimulatory, it increases the PO by even more in the case of F508del. So where does MANT bind? So it turned, well, MANT is binding to the ATP binding site. What is it doing? So this is a top view of the NBDs. I'm going to show you a bigger expanded version. This is the next slide. Sorry, this is the top view up here. So MANT is binding probably to both of the sites, but we only see the benzoic acid derivative on the ribose clearly resolved in one of the sites. And the next slide will show you why that is. But the bottom line, this is a symmetrical NBD, and it tends to favor also this very more closed state of the transmembrane domain. What the MANT does is it torques open. It basically clamps the site with the MANT, the two domains clamp down on it, and they rotate open, and they produce an asymmetrical state. And the next slide is going to show that much more clearly. So this site has the Walker A NBD. The open site has the degenerate Walker A motif from NBD1 and the degenerate signature sequence motif from NBD2. That'll also be clear in the next slide. And then here's just an expanded view of this that shows the MANT group that's well ordered is tightly tucked in this little cavity at the NBD interface, and this site is very clearly peeled open. And so the benzoic acid ring is in a little cavity here that I'll show you another view of in a moment. I'll go to the next slide. So again, there's a degenerate ATP binding site, right, because it has a Walker A that has a serine where it should have a glutamate, and then this should be LSGGQ, and it's LSHGH. It's always been known as degenerate. It's been known for a very long time. This site is catalytically inactive. And what's shown here is in the gray is what we get the ATP in the canonical site, the symmetrical state, and orange is the MANT. And the colored structures here are the MANT. And so this is a surface view, and this will give you an idea of what's going on if the animation works. So this is the transition between MANT bound and just regular ATP. And so there's, about an 8-degree rotation between the two domains, and it's basically clamping down on the MANT group more tightly. This is the same view from this is now a side view of the same phenomenon. And hopefully this animation works. And it is, this shows you another view of how the MANT is causing the NBDs to clamp down in a different way. Okay, and so this cavity is larger than just the MANT, and so this does look like a druggable site, and I showed you that the MANT increases the open channel probability quite substantially. And so what does it mean about mechanistically? Well, again, the non-canonical asymmetrical inter-NBD conformation that's stabilized by MANT that I just showed you, it favors an alternative conformation in the extracellular exit portal. So there is the transmembrane helices from TMD1 are shown in blue, TMD2 are in green. You can see the TMD1 helices are almost unchanged. They're almost unchanged in all of our structures, but there's conformational variability in TMD2, and in particular, TM12 and TM8 undergo a concerted motion. And the MANT bond state of the NBDs favor that shifted state of the exit portal region. And so that's explaining the population change here in the net movement of it into this asymmetrical state with it's called open. I'd say we're not really sure it's open. It's asymmetrical in the NBDs. It maybe is more open in the extracellular exit portal region. I'm going to show you some pictures of that in a moment. With that. So now this is with the G551D story. So super quickly, we studied G551D because specifically it does not open without a potentiator. We could not see any effect of VX770 in our experiments. So we focused on something hopefully that a construct where we would know whether the potentiator was working. And VX770, and then we found all the conformations in the samples. Every circle here is the area of the circle is proportional to the percent of the population of these different structures that are shown. These are actually electron density maps shown up here. So when we add VX770 here, nothing happens at all. But on the other hand, when we add VX445 together with VX770, we get a new conformation. And with G551D, the NBDs you can see are now so this is actually the reference state that I just showed you. Here with the VX445 added, you can see the NBDs are much closer, but they're not as close as they are here, and I'll show you that more carefully in the next slide. So exclusively with the, and we have not, I should say, tried VX445 alone. Again, we've tried VX770 alone, it does nothing. The combination produces a new state where the MBDs are close to one another, but they're not fully associated. I'll show you that now more closely here. These are this is the inactive state with the MBDs far apart. This is the reference state. The VX445 and VX770 produces what we call the N3 state. You can see it has an internal channel. The internal channel is rather similar to the canonical from the fully symmetrical wild-type structure. The NBDs, you can see, however, cracked apart a bit. And if we now look at the top view of this, these are how the MBDs look in the fully separated state. This is the symmetrical state. Here you can see this is once again an asymmetrical state. And notably, the G551D mutation is down here. So the mutation that's producing electrostatic repulsion is the one where this is closely associated. And the important point of that is that the form of the asymmetry here, the more closed site is the site with the mutation. The mutation should keep this site open, but the protein, when it's going into that associated state, puts that site close to, it almost closes it despite the electrostatic repulsion. And that says that this particular asymmetrical state with the fully canonical ATP binding site close and the degenerate site open is clearly favored and clearly what's present in the G551D structure. So I'm almost done. So the VX455 activated conformation of G551D shows a similar non-canonical inter-NBD conformation to that stabilized by Mant. Then we can now compare the transmembrane domain in a couple of these structures. And you can see here, so this is the Mant experiment. This is the G551D experiment. You can see there are two different variants, the transmembrane domain here and here. If we compare the symmetrical versus asymmetrical, now I've just superimposed the gray on this side and the green here, right? The gray and the green are clearly different. But the green, this is now the exit portal structure in green here, matches very closely here. The one here and the grays also match. So they're really two discrete conformations of the exit portal region. The next slide, which is essentially the last one, shows the cavities, internal cavities in these structures. In the completely separated NBD state, you can see that the cavity only really does not go very far. It's really sort of a big trough up above this site. In the completely symmetrical state, there's certainly a cavity, but near the exit portal region is quite occluded. As we go through these asymmetrical states, you can see things are changing down here. And this state actually is something that is the most open-looking state that we've seen. This is an asymmetrical state of the G551D protein that I showed you another view of last time. So that pretty much wraps it up. That's the story. And thank you again for the invitation.
Thank you, John. We have time for one or two questions. Martina?
Tremendous amount of work, nicely summarized. So you showed very clearly how the MBDs and the TMDs change their conformation. And I know it's not your focus, but I keep wondering, is the pathway of the anions like chloride and bicarbonate well defined, or is it still speculation?
I think that this... So the interior part of this channel changes, but very slightly in the different conformations. Let me go back to that slide. It's just it's a big wide well really in the upper part of the channel. Through most of the channel, it's just a very wide well. Sorry, yep, very wide well in here. Anions typically anion channels. Cations, because they're small, they're very strongly hydrated, right? To get rid of the water, you need to have specific binding sites to pass the cations to mediate the dehydration of the cations. Anions, they're much bigger. They're generally not, they're nowhere near as strongly hydrated. They're generally not well-defined anion binding sites in anion channels, so they can be a little bit hard to see the channel. The constriction down here is very small in the state that you see, the dominant state that you see in any structure. And I think the question is whether that's roughly the right one and the dynamics are letting the chloride pass. The electrophysiologists I've spoken to have the anion channel electrophysiologists I've spoken to know the most about it, think that this is probably too small to be passing iodide, but it's not entirely clear. It could be it's dynamic enough that the iodide could pass. And so we've just started collaborating with someone who does electrophysiology, molecular dynamics calculations of ion conductance. In the goal, what he's about to do, Imad Takorji, who's an expert in this area, he's going to compare computationally estimate the conductance of chloride versus iodide, because we figure that's the best. These are not perfect methods, but we figure if we have the right, really the open state, and he will do it, I should say, during a molecular dynamics trajectory. He will calculate the conductance during the trajectory, and that's how we're hoping to get at whether, it's really just a question of dynamics or whether it could be true. We're in digitonin, we're not in natural membranes, we still may be missing the exact open conformation.
Excellent, thank you.
Okay, thank you.
Thank you again.
All right, thank you, John. Next, we would like to invite up our next speaker, Jean-Pierre from University of Cincinnati and Cedars-Sinai. And we're using the laser pointer in PowerPoint here. And you can advance with this.
Good morning. Today I'll be talking about the mechanism of CFTR modulation on endothelial mechanosensitive channels. We have nothing to disclose. So as we all know, the main cause of mortality in CF is still respiratory failure. And as such, the vast majority of studies have focused on the airway epithelium. However, CFTR has been shown to be expressed on other cell types, including endothelial cells. So what really pointed this study here is this figure, which shows the lung vasculature in 2 CF patients. And this is through CT scans and morphometry analysis. And pretty much we reconstructed the vasculature of CF patients. And what we observed was that there was a clear redistribution of the total blood volume from smaller blood vessels that you see here in blue in the healthy CF patients. And when you look at the CF patient with severe lung disease, we see that the majority of the blood is now contained into larger blood vessels. This means that the small pulmonary blood vessels are actually being lost. And with this remodeling, we did some experiment and looked into it further, and even Trikafta cannot bring those vessels back. So once this remodeling happens, there's no going back. So the best course of action here is early intervention. So prevention is what we're trying to achieve here. So our goal with this study is trying to figure out exactly how this process happens. So when you talk about blood vessels, there's typically 2 things that can lead to the loss of the blood vessels. One of them is impaired angiogenesis. So angiogenesis is the formation of new blood vessels from a pre-existing blood vessels. So typically, if you have a blood vessel injury, you try either to repair that vessel. If you cannot, you try and make a new one to just take over. And we suspected that maybe angiogenesis was impacted in CF, so we decided to look into it. So we performed tube formation assay, which is the angiogenesis assay, and we found that inhibition of CFTR led to faster breakdown of the angiogenic tubes. So we started looking at key regulators of angiogenesis, and we stumbled upon this channel called piezo-1. And what piezo-1 does in angiogenesis, it pretty much helps with tip cells pathfinding. So when you have the blood vessels, to make a new one, the endothelial cells have to migrate out of the vessel and then try and create a new path to make a new blood vessels. And this process requires elongation and retraction of those tip cells, and that requires calcium entry. And that calcium entry happens through piezo-1. And other things that piezo-1 are also involved in are shear strength sensing, migration flow alignment, and apoptosis. So we thought maybe CFTR is negatively impacting the function of piezo-1. So we use patch clamping techniques, and we knock down CFTR in the microvascular endothelial cells. And then we try activating piezo-1 with a specific activator called YODA1. And what we found was when CFTR was knocked down, the response to YODA1 of PSO1 was completely abolished. So that was very interesting. So we decided to see if there was a dependence of PSO1 function, if PSO1 function was dependent on CFTR expression. So we co-expressed PSO1 by itself or co-expressed it with CFTR. What we found was when CFTR was present, the response of PSO1 was much higher. We were also curious if this was happening because PSO1 is a cationic channel and CFTR is a chloride channel. So we thought maybe the movement of ions might be playing a role into this. So we co-transfected PSO1 with other chloride channels like TME-M16A. And what we found was that only CFTR had that enhancing effect. So what we did was to know if CFTR needed to be in some sort of proper conformation, we co-transfected with delta F favorate. And what we found was that this mutant didn't enhance PSO1 function. But when we corrected it with CFTR correctors, then the enhancing effect came back. This means that you need to have some sort of wild type like conformation to be able to enhance PSO1 function. So looking at that, because of the conformation dependency, we thought that maybe there could be a direct interaction between PSO1 and CFTR. So we did co-immunoprecipitation. And what we found was that both CFTR and PSO1 co-immunoprecipitated together. So the last thing we did was to co-transfect both PSO1 and wild type CFTR, but this time we decided to also inhibit CFTR. This was to know if both CFTR expression and function were needed. And in fact, when you do inhibit wild type CFTR, the enhancing effect also disappears. So you need both expression and function of CFTR to enhance PSO1 function. Another thing that we also found was we did some bulk RNA sequencing, and we found that gene called KDR, it's VEGF-R2, so it's the main protein, it's the main receptor for angiogenesis, and it's significantly downregulated when CFTR is knocked down, which means that the VEGF-R2 signaling pathway could be impacted here. And if this is the case, then you cannot have proper angiogenesis. So those two might be playing a role into why angiogenesis might not be happening in CF. The other aspect so as I said, there was two factors. The other one is loss of endothelial barrier integrity. So this has to do with the fact that the endothelium forms a semi-permeable barrier, and that's to prevent leakage outside of the blood vessel. So if that barrier is compromised, then you have things like edema. So we decided to look into this. So we took a different approach for this. We use a long-on-the-chip model using a microfluidic device. So what we do is we try to mimic the blood vessel. So we seeded the microvascular endothelial cells directly on the microfluidic chip. Then we connected it to a peristaltic pump. And then we run the flow. What we also did was to have a static or no flow control that we use to make sure that there's no cytotoxicity with all the different agents that we're using in the experiment. What we found was in static condition, using a CFT inhibitor to inhibit CFTR had no adverse effect on cell viability. However, when we introduced flow, there was a sharp reduction in cell viability. So we decided to look into this process. What we first did was to do some live recording of this process. So we have this really fancy microscope that we can use to take images in real time. So we don't have to do it just as you can do it for hours, even weeks. So what we found was that it looked like the cells were pulling away from each other. So with this, we thought, okay, this looks like something that has to do with adherence junctions. So adherence junctions are what keeps the endothelial cells together. So we decided to stain for VE-cadherin, which is the main component of adherence junctions. And we found that when shear stress was present and then you knock down or inhibit CFTR, you see a reduction in VE-cad expression. So based on that, and we just use logical process, we were able to come up with this model here, which is how exactly this happens, and I'm going to walk you through it. So the first thing was, as I mentioned, adenine junctions expression was reduced. And we also, using our bulk RNA sequencing data, we also found that ASR2, which is a gene involved in the synthesis of a lipid called S1P. So S1P is a signaling sphingolipids that increases adenine junction expression, and that gene was reduced. So what we tried was, okay, let's increase S1P level in the cells. To do that, we inhibited S1P lyase, which degrades S1P. So once you inhibit S1P lyase, S1P doesn't get degraded, so it just accumulates inside the cells. And once we did that, we were able to rescue the cells. So as you can see, there's much more cells on the platform. And this would indicate that, in fact, adenine junction disassembly plays a role in this mechanism. Then a key regulator of adenine junctions in endothelial cells is a protease called calpain. So calpain is calcium dependent, and it just cleaves adenine junctions. So we were trying to see, okay, if adenine junction expression is being reduced, maybe that's because calpain activity is actually increased. So we measured calpain activity in the endothelial cells, and we found that, in fact, it was actually increased. So what we did was we inhibited calpain activity using an endogenous inhibitor called caplestatin, and that also rescued the cells. So now we know, one, adenine junctions are being disassembled, and that's happening through calpain. Because calpain is a calcium-dependent protease, we decided to look into intracellular calcium levels. So we use a calcium indicator called FluO8 and loaded our cells with it. And then we looked at what the intracellular calcium level looked like at baseline level. We mean there was no flow present, and it was very low. Once we introduce flow, it shoots up. And then we only did the flow for 1 minute, and then we removed it to allow the calcium to kind of like go back to baseline level. What we noticed was when CFTR was knocked down, that increase in calcium was sharper and also stayed like high for longer. So this led us to believe that there might be some sort of mechanism-sensitive channel that was involved in this process. And that had to be one mechanism-sensitive channel, but also permeable to calcium. So through a process of elimination, we thought our candidate was TRPV4. So we did try blocking PSO1, but this had no effect. But when we blocked TRPV4, we're also able to not only rescue the cells, but when we stained for adenine junction, we also found that the adenine junction integrity was maintained. So now we have adenine junctions being cleaved by calpain, and calpain is being activated by calcium, and then that calcium is coming through TRPV4. So the other question that was left was really, why is TRPV4 being overactivated in the case of CF? So we know from literature that the mechanism-sensitive channels, they're expressed in the cell membrane, and the membrane is a lipid bilayer. So those channels are actually gated by bilayer tension. So if you change the composition of the membrane, you can affect how those channels open and close. So we started looking through literature, and then we found that there was an increase in electronic acid and EPA. Those are two lipids. And what's interesting about those lipids is that they actually can sensitize TRPV4. So actually that channel activates more easily. So we measured electronic acid and EPA in endothelial cell, microvascular endothelial cell, and we found that it was actually increased. And what we did next was to supplement the cell culture media with both electronic acid and EPA. And when we did that, we saw the same effect that CF inhibition had. And then we inhibited TRPV4, and again, also was able to rescue the cells. And the other thing we also did was looking at membrane fluidity. So membrane fluidity, the more fluid the membrane is, the easier it is for that membrane to kind of bend, and the easier it is to activate those channels. When we measured it, it was also, the membrane was more fluid when CFTR was knocked down. What we did next was making sure that the supplemented arachidonic acid and EPA was indeed going to the membrane. So we did some click chemistry just to double check that, in fact, it was going to the cell membrane, which it was. So we proposed that the endothelial cells themselves have a higher metabolism of AA and EPA, but also because it's reported that arachidonic acid is elevated in the blood of CF patients. This means that those cells can also pick up arachidonic acid from the bloodstream and also incorporate it into the membrane. So this could have clinical relevance because you can use a lipid test to see if those patients have like higher risk of developing barrier failure. So this is in summary. In the healthy endothelium, we think that both CFT and PSO are interacting at the cell surface. We don't know by which domain yet, but in the case of CF disease, we think because the protein isn't present, so that interaction is actually reduced. But we also think that there is an increase in AA and EPA levels, which eventually sensitize TRPV4 channel. So this leads to a calcium overload and to overactivation of calpain. And then calpain then goes on and cleaves adherent junctions, which create gaps in the endothelial monolayer. And we think it's both impaired angiogenesis and loss of endothelial barrier integrity that leads to the loss of small pulmonary blood vessels. And I would like to acknowledge the entire NERON lab and also the UC and University of Cincinnati for RNA sequencing and Vanderbilt for lipid analysis. And this work is funded by the American Heart Association pre-doctoral fellowship and the Cystic Fibrosis Foundation. And
Thank you, Jean-Pierre. Any questions?
Hi, Viral from MGH. Super interesting work. I was thinking about the first talk and your talk. I wonder if you could use G551D CFTR to help tease apart the effect of CFTR and piezo and how it's modulating it. Since we learned, or at least I learned, that a VX770 doesn't change the conformation. I wonder if that could be one way to get at this.
yeah, so we've been trying to look into G551D, and we've been using VX770, and this hasn't worked. So now after seeing this, I was actually taking pictures. I'm like, my God, that's why it's not working. So yeah, because only 1 out of 10 cells would like recover the CFTR function. So we haven't been able to like make sure if that was having like an enhancing effect, like when we're rescuing delta F. With G551D, we're still not sure. So we're trying.
Gotcha. But probably then it suggests it's more of a conformational change than ion transport.
Yeah.
Interesting. Cool.
Thank you.
So you showed that there's a co-IP with CFTR, and then on your model, you kind of indicated that the R domain is interacting. Have you actually looked which part of CFTR is involved in this interaction?
No, so as of now, we don't know. So that's something that we're planning on looking into.
And then I'm also wondering, when you have a delta F that sticks to the endoplasmic reticulum, whether this interaction occurs and your protein doesn't make it to the membrane, or whether it's not affected. That would be interesting too.
Our guess is that the PSO1 function is probably reduced. So it's not
Would its intracellular localization be affected? Would it be
for PSO1?
Yeah.
Oh, that's a very interesting question. That's something that we haven't looked into. My guess would be that it's a 50-50 guess, because CFTR might well be stabilizing piezo on the cell surface. So it's very possible that that could be happening.
Thank you.
Thank you very much for your presentation. In endothelial cells, but also in many other cells, mechanosensation is often linked to the release of nucleotides and signaling in a paracrine fashion. And as you manipulate with your cells with mechanical stimuli, is there a reason to believe that these signals, which are very lively in other systems, could play a role in your system? Neuronergy signaling is the question.
Yeah, I don't have the answer to this question because that's not even something that we really looked into. I wish I had a better answer.
And we have a question from online from Ines Pankanin. Have you checked piezo channel expression in epithelial cells?
Yes, we did. So both with the bulk RNA sequencing and also qPCR.
Yeah, very nice presentation. Do you have any evidence that this is happening in vivo? And if so, is this only happening in the endothelial cells of the lung or in other organs and tissues too?
So for in vivo, so that didn't come from us. So there's a study that was published last December, and they were looking into pneumonia. And what they found was that CFTR expression was reduced in pneumonia. So what they did was they used rats and mice and they perfused the lung with CFT inhibitor. And when they did that, they also saw pulmonary edema. And then they also inhibited TRPV4 and that fixed the problem. So it's most likely that the same thing is also happening in vivo. If this thing is only compartmentalized to the lung, my guess would be no. But again, we've only been looking at the lungs, so I have no way of knowing for sure if it's just the lungs or any other tissue. But PSO1 is involved in renal physiology and all those things, so it's very well possible that those can also be affected.
Thank you. I think that's all the time we have for questions. Thank you. And while I invite up our next speaker, if those in the back would like to make your way up towards maybe the second or third row, there's plenty more seats up here. So please take the opportunity to do that. Or if others are sitting on the outside, wouldn't mind moving in, that would be great. Okay, so next I'd like to welcome Carrie Thrasher from UAB. Let's get your laser pointer going.
Thank you to the moderators for inviting me to speak today and everyone for coming out on the last day super early in the morning. Okay, so I'm going to be talking about a compound that we published a couple years ago as a novel read-through agent, but my project specifically focuses on its mode of action, how it's working. Okay, so here's my disclosure slide, nothing. Okay, so as I probably don't really need to show this at this point, we've seen it a lot at this meeting, but I mostly focus on CF caused by nonsense mutations. So far, around 150 different CF causing PTCs or premature termination codons have been identified in CF patients. So about 10%, according to the CFTR2 database, of alleles that cause CF are nonsense mutations. And so while this is a small percent of CF causing mutations, the other around 90% are eligible for the highly effective modulator therapy like ETI, but that therapy works on the level of the CFTR protein. And in the case of nonsense mutations, once you export a PTC containing mRNA transcript to the cytoplasm, it's subject to degradation by nonsense mediated mRNA decay or NMD. The remaining transcripts that survive NMD can then be translated, but translation will terminate prematurely at the site of the PTC. And so you'll really release a low amount of truncated protein that's usually non-functional and rapidly turned over. So there's really no protein substrate for ETI to work upon for patients with nonsense mutations. Okay. So the therapeutic option that we're really focusing on in the Bedwell lab is called translational read-through. This does happen endogenously in the cell at very low levels. So I just wanted to walk you through how this occurs. So you have the eukaryotic release factor ERF1 and its GTPase ERF3 shown here. And you can have a near cognate tRNA. So while it's not going to be perfectly cognate to a stop codon, it will match at 2 of the 3 nucleotide positions. So normally 99.9% 99% of the time, you're going to have the release factors come in, bind to the PTC, and you're going to have release of a truncated non-functional protein. However, a very small percent of the time, you can have near cognate tRNA incorporation right here and continue translating in frame for the remainder of the coding region. So our aim here was really to perform a high throughput screen to identify novel small molecule compounds that increase the frequency of this translational read-through to occur over the endogenous levels. So to that end, our lab, in conjunction with Steve Rowe's lab at UAB and Southern Research in Birmingham, performed a high throughput screen of over 750,000 small molecule compounds. And one of the hits that came out of the screen is this molecule called SRI-41315. So it does induce read-through. I'm showing here on a read-through reporter cassette, a dual luciferase, but if you can check out that paper to the side there, that's where we show read-through of CFTR and CFTR activity also. And it does this without causing a significant cell toxicity. This is an ATP assay here, but I've also looked at various caspase assays as well. So it seems like it's not that toxic to the cells. Okay, so back to our schematic of read-through and how this occurs. So sort of one of the first hypotheses that we had as to how we might be increasing the frequency of this occurring is just by reducing the levels of the release factor protein in the cells. So you can have more efficient competition by the near cognate tRNAs. And that is what we see when we look at ERF1 protein levels over time, shown here. You get reduction in steady state ERF1 levels, and this was also shown by the CFTR lab as well. Okay, so if we're getting rid of the protein, we wanted to see how we were getting rid of the protein. Is it proteasomal dependent? So we use the proteasomal inhibitor MG132, shown in blue, and compared to a control without the proteasome inhibitor where we see ERF1 degradation, we see non-significant degradation of ERF1 when you block the proteasome. So now we know it is proteasome dependent. Since ERF1 is obviously involved in translation, we wanted to know if this degradation was dependent on its function. So does it need to be involved in translation to get degraded, or can it just be in the cytoplasm and get degraded there? So we use the translation inhibitor anisomycin, shown in blue, and blocking translation rescued ERF1 degradation compared to the vehicle control shown in gray. So it is translation dependent. So since we know that this is happening during the mechanism of translation, we did a RiboSeq analysis. So just a brief explanation of the technique for those of you that are unfamiliar with it. You can freeze the ribosomes where they are translating across every mRNA in the transcriptome and pull down basically the ribosomes and RNase treat. So you basically degrade all of the RNA that was between the ribosomes, and once you free the mRNAs from the ribosomes, you have these what we call ribosome protected fragments that you can map to the human transcriptome to sort of get a count of where every ribosome was at the time that you added the translation inhibitor. So this also answers everyone's favorite question: is are you reading through normal termination codons as well as premature termination codons? So here's the answer. So to start with, we can map a set of the reads to the human transcriptome and then align them based on whatever motif or sequence feature along the transcript that you want. Here along the x-axis, we have them aligned relative to the stop codon. So this is transcriptome-wide relative to the stop codon. And you see this large peak at stop codons that we always see with RiboSeq, just because translation termination is a slower process than elongation. So you're going to have more reads at those areas. If you see in black, that's our vehicle control, and we use G418 control on this as well. That's a very potent aminoglycoside read-through agent that we use in the lab. In gold, and then our compound in light blue. So you can see here the peaks in black from the DMSO control are what we would consider normal. And then G418 control actually lowered that peak, so you had less time spent at stop codons for the G418 control. However, with our compound, we went the opposite direction. We had more time time spent at stop codons with our compound relative to the vehicle control and the G418 control. So this kind of makes it look like our compound is causing ribosomal pausing at the stop codon. So if it kind of makes sense, if you're reducing ERF1 levels, you're spending more time at that position, okay? And then we can zoom in the 3' UTR region downstream of stop codons. Again, this is transcriptome wide, so these are normal termination codons that we're looking at right here. And you can see in black is our DMSO control, and gold is the G418. So we actually do see an appreciable increase of ribosomes in the 3' UTR with G418, and you see these like peaks that are happening right here. That's what we call nucleotide periodicity. So that's indicative of actively translating ribosomes. So not only are there ribosomes in the 3' UTR with G418, they're still translating. But with our compound in blue, it matches the DMSO control, and we see no nucleotide periodicity there. So we're not really seeing appreciable read-through of normal termination codons with our compound. Okay, so I'm going to pause here and sort of recap what we just talked about before it gets too complicated. So we have a ribosome that reaches the PTC right here, this first ribosome. You have ERF1 that is coming in and actively binding to the stop codon. It's entering the A site. This is causing our ribosome to stall. Also, we saw ribosome stalled at the stop codon, and that's never, it's not a static system where you have one ribosome per message. You're always going to have another ribosome coming along behind it also. So I added a trailing ribosome right behind it, and then we also know that ERF1 is somehow getting degraded via the proteasome after that. So that's what we've seen so far, and now we're going to add to that model. Okay. So around that time, a new paper came out earlier this year where it's a different compound called ternatin 4, degrades a different translation-related protein, EEF1A. It's an elongation factor, but that protein also enters the A site in the ribosome during translation elongation. And they found that this compound causes the ribosome to stall with the elongation protein in the A site, causing ribosome collisions shown here. And that's recruiting E3 ligases to degrade that protein via the proteasome. So this sounded very similar to our compound. We're degrading two different proteins. The structures are completely unrelated between the compounds, but it sounded pretty similar to ours. So I ordered siRNAs to the two different E3 ligases they reported in their paper. And so the first one, RNF14, when I knock it down, shown in blue, I rescue ERF1 degradation that's seen in gray with the scramble control. And the same thing with the RNF25 knockdown. I rescue ERF1 degradation seen in the gray scramble control. So this seems like this is a conserved sort of ribosome quality control mechanism. If you stall the leading ribosome at the stop codon for too long, that you have ribosomal collisions from the trailing ribosome, you're going to recruit these same two E3 ligases to clear whatever is stuck in the A site so you can free up the collision. Okay. So we are a translation lab, so ribosome collisions were not new to us. This has been a pretty hot topic in the field of translation for the past 5 years or so. So there was a breadth of work already done on ribosome collisions for us that saved us some time. And we also knew some of the consequences of ribosome collisions for this. So if we're causing ribosome collisions, there's possible stress response pathways that can be activated as a consequence of ribosome collisions. So the two different stress response pathways are the integrated stress response, or ISR, which is sort of an intermediate stress response pathway. This is indicative that the cells are going to survive, be able to clear the stall, and then the cells are going to be okay. And then there's a more severe stress response pathway, the ribotoxic stress response, or RSR, shown on the bottom. It's known to be a more severe stress pathway that you activate MAPK signaling and ultimately apoptosis if it's retained in that stress state for too long. And so we decided to look at both of those to see if our compound is inducing either the integrated stress response or the ribotoxic stress response. So for the first one, the integrated stress response, just as an overview for those who are unfamiliar with it, there's 4 different protein kinases that can activate the phosphorylation of EIF2A. The one that we're interested in is GCN2, which is known to be a sensor of ribosome collisions, and that's been published. So GCN2 induces EIF2A into phospho-EIF2A, which results in the translation of ATF4, which then becomes a transcription factor into the nucleus for a certain subset of genes to be upregulated. And we call these ISR target genes. They're only expressed under the condition of the integrated stress response being induced. So I'm looking at showing two of these right here, CHOP and GAD34. So you can see in gray where I add 41315 and a scramble control, I get induction of these ISR genes. So it looks like we are inducing the integrated stress response with this compound. And this is also GCN1 dependent, shown in blue. When I knock down GCN1, you see a significant decrease in the expression. So it seems like what's been published previously with the ribosome collisions inducing ISR is happening in our case. However, this is a transient effect. I did bulk RNA-seq over time, and this induction does go down after about 12 hours. Okay, next we're going to look at the ribotoxic stress response. Again, this is the more severe stress response pathway that may ultimately lead to apoptosis if prolonged. And like I said, there's a different protein sensor on the side of the ribosome collisions called ZAC-alpha. Upon ribosome collisions, it triggers the MAP kinase pathway, ultimately resulting in the phosphorylation of this protein called JNK, which can lead to apoptosis. So to start with, I looked at the expression of total JNK. Are we changing the expression of total JNK? And it doesn't appear that we are. However, the phosphorylation state of JNK is increasing over time, peaking again at 12 hours. It matches the integrated stress response bulk RNA-seq that I did. However, it does return back to baseline over time. So this kind of coincides with my cell viability assays. I did not see apoptosis through various cell viability assays. So it looks like any kind of stress response that we are activating are transient in nature and is coming back down to baseline over time. So this leads me to the final mechanism, and I know it's complicated and a lot of proteins on here, but basically once a ribosome encounters the PTC and the ribosomal A site, ERF1 comes in and binds. Our compound causes it to get stuck in the ribosomal A site, which makes this leading ribosome stall. It can move forward. The trailing ribosome will come along and crash into it, causing a ribosome collision. This then recruits these ribosome quality control E3 ligases, RNF14 and RNF25, to ubiquitinate ERF1 and target to the proteasome. However, there are ribosomal collision sensors present in the cell, like GCN1 shown here, and ZAC-alpha, that will induce the integrated stress response and the ribotoxic stress response in a transient manner that does return back to baseline over time. And with that, I just want to acknowledge some people in my lab, the CF Research Center at UAB, specifically Steve, Carl, and Jay, who have done a lot of work with the compounds that we discovered in the screen. Southern Research Institute, who is the chemistry team that we work with. The CFFT lab, who did a lot of the initial mechanistic studies for 41315 that led to my project. Our funding sources, and Rachel Green's lab at Hopkins for the RiboSeq data. And I also want to point out that this is not the only hit we have from the screen. You can visit Steve and Jay's poster if it's not down. If not, email them, I guess. I don't know. 318, we have some novel ERF3 degraders that are more potent and more efficacious than the compound that I just talked about today. And 271, Ming Du, she works on combining different read-through molecules together based on their mechanism of action. And I'm ready for any questions. Thank you.
Thank you very much, Carrie.
Great. This is really beautiful, Nick. Very exciting story. I'm curious about the direct mode of action of the compound. So you showed a model where it's directly binding to ERF1 and sort of getting it stuck in the ribosome. Like, can you say a little bit more about that, or could there be other modes of action?
It's binding in the A site of the ribosome, not to the protein directly. Yeah, so it's just kind of interesting because it is binding directly to the A site of the ribosome, but we did look at other proteins that enter the A site, like the EEF1A protein that I talked about before, and there's no change. So it's somehow still binding to the A site, but specific to ERF1 and not other proteins that enter. Yeah.
Okay, great. Thank you.
Thank you.
I have a question about if you have noticed a dose dependency with the degree of knockdown that you get with ERF1, and in turn effects on read-through and or rescue of nonsense CFTR variants.
Yeah, so when we do a dose curve or a time course, however you can put it, definitely the lower ERF1 gets, the more read-through you're going to get. It's kind of important more in the ERF3 degraders, because at some point with the ERF3 degraders, you destabilize ERF1, and it's only when ERF1 is gone do you get read-through. So here we don't really change ERF3 levels, and we're still able to get read-through at lower doses and everything. But it seems like ERF1 is like the important one. Even in the ERF3 degraders, it's still ERF1 that's the important one.
Hi, that was great talk. A lot going on there.
Yeah.
So just thinking about read through, so you're enhancing read through, but your drug binds to the A site.
Yeah.
And so this is not how I would have thought this was going to work.
Yeah.
So how's it working?
Yeah. I had the same question. If you have an occluded A site, how's the tRNA getting in there? Yeah. I think it's sort of what I was telling Catherine is that it's going to be sort of a temporal thing. You have to reduce the steady state levels of ERF1 enough that you're going to have an empty A site at that point. So once you've caused the occlusion enough times and ubiquitinated and degraded enough ERF1, then all of a sudden all of your A sites are going to be empty for read-through.
So if you do bulk RNA-seq, do you see a reduction in transcript levels? Because this also will degrade the mRNA in which these ribosomes are bound to. Is that true?
I did bulk RNA-seq. I didn't look at since it's bulk, I didn't have anything obviously with a PTC that I could go in and look at and everything. But I mean, I might be able to try to find something with like a UORF or, something like that I could see.
And then just the last question, did you combine your G418 with the degrader and then do ribosomal profiling to look to see NTC read-through? Does that make sense?
We haven't done that.
Okay. Yeah.
I mean, I expect, let me put it this way. You can use way less G418 with ours. So at like an EC10 of G418 plus the ERF1 degraders, that would be interesting to look at because you might reduce NTC read-through by using only an EC10 of G418, but still get a crap ton of read-through.
Yep. Yeah. That's a quantitative measure.
Yeah, crap ton. Yeah.
Thank you. Really nice talk. I just want to clarify that stress response you shows from the native stop codon, not PTC. Is true?
The integrated stress response?
Well, two types of stress response happens at PTC or
the collisions, it'll be everywhere. We haven't done polysome or anything, but whenever you do polysome, you're going to be looking at all ribosomes. But it's going to be not just at PTCs that you're going to cause ribosomal collisions. Anytime that ERF1 gets in the A site, it's going to get stuck.
So do you have any good idea why there is no stop codon read-through?
No.
Native stop codon? I mean, is there a big difference between the PTC versus NTC?
Why we get read through it at PTCs versus NTCs.
Yeah.
I mean, I can hand wave it as much as you want as far as, the distance from PAB of a normal termination codon versus a premature termination codon. If you looked at G418, you saw it like trend down, even in the 3' UTR. That's from multiple tandem in-frame stop codons. But no, I don't have like a good reason why, it seems to read through PTCs that we see. We've seen it not just in the reporters, but in CFTR. But the RiboSeq reveals no read-through with the NTC. So I don't know.
All right, thank you.
Okay, and last question.
Hi, hello, thank you. Very impressive. And linking to this question, do you think that there are positional effects of different correction of different mutations falling in different positions?
Yeah, I mean, it's going to be a bunch of different ones depending on the stop codon because it's not just positional, like how far away from the PAB or the normal termination codon is. It's going to be completely context specific. Different stop codons read through at different efficiencies, and even outside of the context, nucleotides upstream or downstream from the stop codon will affect how a drug reads through. So it is going to be position dependent probably, but it's also going to be stop codon dependent. It's going to be nucleotide dependent for the native sequence and everything.
And also with the other mechanism, for example, non-sense-mediated decay inhibition, for example.
So basically, we use NMD inhibitors as like tool compounds, but we've seen that if you have enough efficacy in your read through, you inhibit NMD just by not triggering it because you're not terminating prematurely. So, it's just one of the ways that it would be very toxic to give someone an NMD inhibitor and everything. But if you read through, you don't trigger NMD, because it's usually triggered through the pioneer round of translation. I know we always show it NMDs first and then translation. It's actually translations first. After the pioneer round of translation, you trigger NMD. So if you read through during the pioneer round of translation, you're not going to trigger NMD.
All right. Thank you again, Carrie. All right. Next, I would like to welcome Christina Barilla. Also, you're at UAB? Yes? Yeah. Are you at UAB? Are you at UAB? Yes. Yes. Okay. Okay.
Good morning, everyone, and thank you for the opportunity to show our work in developing a platform that can be used to study the response of CFTR to modulators. I don't have anything to disclose. As you may be familiar with the latest release of the CFTR2 database, we now have more than 700 mutations that are classified as a CF causing. And from here, we already have 178 that are eligible to treat with the ETI. But the majority, although the percentage may be lower in the population, are not approved to this treatment yet. And these can contain mutation we know or we expect may not respond because they are premature termination codon or some type of splicing mutation. But we also have a lot of just two rare mutations that require further characterization before reaching the approval state. Overall, we know that we have a variable clinical feature in this patient, even when we have the same type of mutation. And we have seen in case of the F508del mutation that even when the mutation is the same, the efficacy of the modulator may be variable. And this variability can be depending on both genetic and non-genetic factor, but particular if we focus on the genetic factor, this will depend on the type of CFTR mutation that the patient has, as well as SNPs that we may not be aware that are in deep intronic region or regulatory elements. But also we have a non-CFTR mutation that may affect the phenotype. Right now, we have a really good model to study the CFTR function, which for the purpose of this presentation, I classified in two groups as a non-patient and the patient-specific. For the non-patient specific, they are really good to study a specific CFTR mutation and its effect on the CFTR biology. But as I mentioned, the patient background, the genetic background is really important, and we risk to lose all this information with this model. And in case of the cDNA-based approach, such as the FRT, we are missing the possibility to model some of these mutations, for example, like the one in the regulatory element. On the patient-specific side, we can carry all the genetic background of the patient, but depending on which tissue are you interested in, if we are studying intestinal organoid, you may have differences in the expression level compared with the airway that we are interested in, so may not be perfectly matching. Or in case of primary cells, we are often limited in the number of cells that we can use for the study. For us, what we would like to have is a patient-specific model so that we can have the patient's mutation, which are often in a compound heterozygosity. We also can carry all potential genetic modifiers with these models, and we want to retain tissue-specific information, and in our case, it's interest on the airway epithelium, and we want to have as many cells as we want whenever we want. So, for this reason, we decided to pursue an IPS or induced pluripotent stem cells-based approach. And this is a collaborative project between different institutes. We have at the UAB George Solomon Group, and at the Emory we have Eric Sorscher, Arlene Stechenko, and Rachel Lineman groups, which are involved in the enrollment of the patient and the sample collection. And then our group, formerly at UT Houston and currently joining the UAB, are responsible for the generation of iPS cells and then the differentiation into air liquid interface culture. At this point, they would be ready for the electrophysiology analysis, and this is performed by Andras Raab in Eric Sorscher group. For the differentiation protocol, we are using basically the same protocol that we published a couple of years ago in collaboration with Finn Hawkins and Darrell Cotton, which focus on the development of induced basal cells, which can be isolated using NACFR and EpiCam as a surface marker, and then plated in air liquid interface culture. The only modification that we introduced is an additional sorting step, which use F3 and EGFR as sorting markers. And the reason we introduced this step is because once we applied our protocol to a larger number of non-reporter iPS cells, we noticed variability in the percentage of on the yield of induced basal cells that we were generating, with some of the line performing relatively less good. But when we introduced the sorting step, we now have an overall improvement of the yield of the cells of interest, and this is particularly important and evident for those lines which were not performing as well from the beginning. And these now allow us to apply the protocol to a larger number of iPS cells that we can now study. Once we have the cells in a liquid interface culture, we now have a model that mimic the pseudostratified airway epithelium, and we confirm that we have basal cells, ciliated cells, and secretory cells in culture, so we can now move on the electrophysiology assay. This is done through the assay chamber analysis, and we usually perform a 48-hour pre-treatment, which is either with the DMSO or modulators, and for this study is mainly ETI. And then we will have a series of acute treatment during the assay, and we record the current change in either in response to either activation or inhibition of CFTR. For this presentation, I'm summarizing the data that come from the difference in culture stimulated from the CFTR-specific inhibitor. And this is one example of the summary that you will see in the next slides. All the data are reported as a percentage of wild-type current, which I'm showing this graph is generated by calling 100% the average of the current measure from the DMSO sample in two different non-CF line. The other line that I'm showing in this graph are from a CF patient with mutation we knew they could or could not respond to ETI. Sorry. And for example, we had this patient which has a splicing mutation and a stop codon we expected would not respond to modulator or really minimum. And we indeed didn't detect any response with ETI. In this patient, we have a frame shift which will result in a premature stop codon, and the G551D, which we know respond to the VX770, which is included in the ETI. And indeed, we observe this response with the ETI. I'm sorry. Similarly, we notice this response with the homozygote F508del patients. These are 3 different patients, and we observe the response in all 3 with a similar magnitude. This was really encouraging for us because one of the reasons that the people usually stay away from IPSS is that the protocol was not reliable enough to guarantee reproducibility. But now we see the reproducibility of the data between different patients and also I'm sorry. But also we see that we can reproduce both the absence of response as well as the presence of response between different clones of the same iPSS, as well as between different experiments within the same clone. So I'm hoping that I convince you that we have a robust and reproducible platform that we can use, which can give us information based on the patient mutation. So now we have ongoing studies that also include patients with mutation. We don't have enough data in the literature to know if they will respond to ETI. And we also have 2 clinical trials involving this platform. Here I'm showing some of the data from this unknown response group. I don't have the time to focus on all of them, but I would like to point it out, for example, this patient that had a stop codon mutation, as well as the V456A, which was approved to use the ETI by the FDA while we were performing this analysis. So this was a further confirmation that we could observe and predict a response based on the genotype. And if you didn't have the chance, I would invite you to visit Rachel's poster, where she has also the clinical information from this patient. Another result that was really encouraging for us was with this patient that had a stop codon mutation as well as N1303K, which we observed a relative big response to the ETI, and this motivated the beginning of a new clinical trial focused on N1303K patients. And George has the poster with preliminary data from the patient in this study. So I hope I showed you that we have a reproducible methodology that give a result with a genotype-specific response, and we can now extend to a larger number of CF patients, which we can include some of even unknown mutation that we are interested in. And we saw when we applied the protocol to a variety of partial function mutation, we can observe a variety of ETI responses, and some of them may be good candidate to start a new clinical trial, like it was N1303K for us. Once all the clinical data will be collected and we will done with the in vitro data, we are going to check if we can have a correlation between the two data sets. And again, those are the two posters where you can see the preliminary data on the patients. Another thing that we are interested in is see if we can also have a reproducibility between different cell sources. This because all the data I've shown you so far are from a fibroblast-derived iPS cells, but we know a patient will be probably more willing to donate blood than a biopsy, so we are trying to see if we can extend this platform to different cell sources. To do this, we compared for just some of the patients, either iPS derived from fibroblast or blood, and we compare the result. Right now, we see that there is some difference in the magnitude of response, but we could confirm the genotype-specific response. And we will perform this in a larger number of patients where we can see if there is a trend in this difference in magnitude, or if it's just because, for example, our control are also fibroblast-derived, like the non-CF. Lastly, we are also interested in see if we can see not just a genotype specificity, but also a patient specificity to the response, which is the main purpose of the personalized medicine. And in this regard, we had a preliminary test from these two patients. They are two siblings that have the same CFTR genotype. And although the relative response to the ETI was similar, we see that there is a basal level of CFTR current in the DMSO state that is a little different. So we may be able to detect something that is patient-specific, but we definitely need to increase the cohort before being able to say that. Lastly, I just want to thank all the people involved in this project at both UAB and Emory, and also our group. Previously at UT, some of them moved to the UAB, and thank you for the patience with my voice too.
Thank you, Christina. Go ahead.
That's a great project, fascinating data. I couldn't help but notice that your responses seem higher than what we see in primary cultures. Do you have any idea why that is? Do you think there might be some regulating factors in primary cultures, or you have here some components in the media that upregulate function, or what are your thoughts about that?
For this, we since it's like a new approach, we started by using the same condition that we use for primary cells, which may overdose with the iPS culture. We don't know that yet. So we are trying to sort of modulate the assaying to see if we can see some differences there. So it may be that there is something also specific just to either the protocol or to the iPS response, or it may be just that the iPS cells are a little more sensitive to detect. That's one possibility.
So I have the same question as Martina, but now I have two comments. First, for the N103K, I can only confirm from the French patient data that we just published that indeed they respond in vitro and they benefit of ETI, and they respond in the primary nasal cell model by 10 to 15% increase of rescue, so more or less in the same range as yours. And the last mutant with the two siblings is a splicing mutations, and so there can be a splicing specific patient regulation. So maybe you could check also. That would be really interesting to check in the nasal cells of the patient, the respiratory cells of the patients, and in the iPS cells of the patients, if you have the same level of Y-type transcript. So that could be really interesting also, proof of concept, what you modify in fact, between the native and the derived iPS cells.
Thank you for the suggestion.
Congratulations, wonderful data. Are you also planning to study bicarbonate secretion in your approach?
We are trying to, for now, just optimize the platform to see if it works. So once we...
Seems to work.
Yeah. Once we complete that, we definitely can use this platform for, hopefully, I don't say everything, but for a lot more information.
I have a question about the percent of wild type currents that you were showing. Was that just a Forskolin-mediated response, or was there also a potentiator there?
For so how we calculated the percentage, that was only the MSO in the CFTR inhibitor current. So not from the Forskolin data, but from the specific inhibition of the CFTR.
Okay, thank you. All right.
Could you fill us in on the potential role of the epigenome and the ability of the iPSCs to completely reproduce the in vivo kind of regulation of CFTR?
We didn't do study on that. I believe Anna Harris was doing some of the preliminary data on that one with a different protocol, so I'm not sure how translatable they are. But in theory, she was seeing step-by-step activation of the CFTR even when it was not actually expressed, so possibly during the differentiation, while the correct cell show up, it's recapitulating also the DNA conformation to facilitate the expression of the CFTR. We just didn't test in our specific platform.
All right. I think that's all the time we have for questions. Thank you, Christina. All right. And then lastly, I would like to welcome Viral Shah from MGH.
Thank you. Good morning, everyone. Thank you to the organizers for this opportunity to chat. And thank you all for coming early on a Saturday morning. Today I'll be talking about label-free imaging. So let's get right into it. I don't have anything to disclose. Let me first tell you why we even approach this. What's our rationale? So as we know, there are many different airway epithelial cell types, and they have varying abundance on the airway surface. So let me illustrate that for you. So here's an airway. You can see the nuclei in blue here with DAPI, and then you can see ionocytes, the CFTR-rich ionocytes, in green. And you can see that if you look at certain regions of this airway, you'll miss the ionocytes. So how could we study a cell type if we can't see it, right? The ionocytes may not even be in our experimental field of view. And so it's critical to be able to identify the cell types in live imaging so we can understand what they do. So this comes with a number of challenges. One thing we could do to identify cells in real time is using genetically encoded fluorescent markers. The challenge here is, though, we have to modify the cells. You can't really do that in vivo in humans. One thing we can do with human samples is we can fix them and stain them to identify the cell types, but at that point, the cells are dead, so you can't really study their function. Another approach is using autofluorescence. This is when you stimulate endogenous molecules to emit light at a specific wavelength. The advantages here is that you don't need to modify the cells, and you can do this in live tissues in all different species, including humans. So the question is, what autofluorescent molecules should we go after to do this? So we looked at autofluorescent metabolic cofactors, specifically NADH and FAD. And what's interesting about this is that NADH is autofluorescent, while its oxidized form, NAD, is not. Conversely, FAD is autofluorescent, while FADH2 is not. And as we know, these molecules are enriched in different processes. In glycolysis, we have more NADH. In oxidative phosphorylation, we have more FAD. Now, those in the cancer field have taken advantage of this because malignant cells preferentially undergo glycolysis, right? That's the Warburg effect, aerobic glycolysis. And so here's an example looking at thyroid papillary carcinoma with NADH imaging, and you can see that malignancy lights up, and it's correlated with pathology. So we thought the airway epithelium, they do a lot of different things, those cells. They have different processes going on. Can we utilize NADH and FAD autofluorescence to distinguish all the different airway epithelial cell types in living tissue? So how can we get at this? Well, first we needed to develop a tracheal explant model. So here we took the mouse trachea, cut along the anterior part of the membrane, unrolled the mouse trachea, bisected it, sutured it onto an O-ring so that we can fix this onto a customized platform where we could keep the tissue alive for days and also do 2-photon imaging of the luminal surface here on top. So let me show you what we found. So first, we did autofluorescent imaging, and this is how you'll see it throughout this presentation. You'll see FAD in yellow and NADH in blue. First, we did the autofluorescence. We registered this location, then fixed the cells, stained with conventional markers, and went back to determine the autofluorescence signature. So this, for example, we stained with acylated tubulin, which stains ciliated cells, and you see those cilia in green. We can go back and look to see the autofluorescence signature, and the ciliated cells they're yellow. They're really rich in FAD. And you can distinguish them from these other cells, these blue cells around them, which turn out to be secretory cells. So here we see CSP staining in magenta, and the secretory cells are blue with these FAD punctate. So just by eye, you can distinguish ciliated cells versus secretory cells. Well, we can quantify this with autofluorescence. That's here, quantifying the autofluorescence here on the y-axis. We use an autofluorescence ratio, which measures the total FAD autofluorescence over the sum. That's just to reduce the light scattering and mitochondrial differences. And clearly here we can distinguish ciliated cells from secretory cells. So common cell types can be distinguished with autofluorescence alone. Now the whole purpose of this was to get at the rare cells. So how can we do this with the rare cells? Well, as with everything with rare cells, it tends to be harder. So let's take a look at the CFTR-rich ionocyte. So here we see the autofluorescence imaging, FAD in yellow, NADH in blue, staining with conventional markers, here BSND to stain the ionocyte, and then here's a drawn overlay. And what's interesting here is you see that the ionocytes have these FAD-rich punctate, but the rest of the cell is pretty dark. The rest of the autofluorescence is minimal. And unfortunately, the other rare cells are just like this. Tough cells, stained here by GNAT3, and neuroendocrine cells, stained by PGP9, have a very low autofluorescence signature. And so using autofluorescence alone here wasn't going to cut it. But, looking at these cells, they're all these interesting shapes. And this could potentially be another non-invasive way to identify the cells. So we thought, could we combine autofluorescence with the morphological characteristics to get at these different cell types? So we looked at nuclear position, X length, Y length, Z length, and you can do this all while you're doing the autofluorescence imaging. We also wanted to get a better spatial characterization of the autofluorescence. What do I mean by that? So let's go back to the ionocyte, one of my favorite cell types, of course. And you see here, there's the FAD punctate. This is a top en face view. Let's look at a cross-sectional view. So this is an XZ optical section. And what's interesting here about the ionocyte is that these FAD-rich regions tend to be towards the top, the apical surface. And so what we can do is then determine a spatial characteristic, a distribution of the autofluorescence as another parameter. This is also interesting from a physiologic standpoint, but for our purposes, we wanted to use this as another determination characteristic. So we quantified and compiled all these different morphological and autofluorescent characteristics for all 7 different airway epithelial cell types, and we have all these 12 different parameters. You don't need to look at this table. Because then the question is, can we use this data to distinguish the cell types? So to get at this, we used unsupervised clustering based on autofluorescence and morphology. Now, this technique is something you're familiar with when you look at single-cell RNA sequencing. So you have hundreds of genes to help distinguish these cell types. Here, we have 12 different characteristics. And it looks like we can do a pretty good job of distinguishing all 7 different airway epithelial cell types. But the question we really wanted to get at is, can we use this data to prospectively determine cell identity? So to get at this, we used machine learning algorithms. So we took our data set, split it into a training data set and into a test data set. So we trained different machine learning algorithms to identify the characteristics of these different cell types, and then tested whether they could prospectively identify this from the data set. And so that's what you see here is a confusion matrix. These are the known cell type identities, and here's what the machine learning algorithm predicted them to be. And you can see it does a pretty good job of identifying the cell types. Now, we thought, well, we have morphology, we have autofluorescence. Do you really need the autofluorescence? Seems kind of cumbersome. So we redid the machine learning algorithm, this time taking out the autofluorescence data and only including morphology. And again, here's the confusion matrix, and you see it does a worse job in predicting the cell types. So you need both autofluorescence and morphology characteristics to be able to accurately predict the different cell types. Now, up until this point, I've shown you native mouse trachea. That's very different than what we see in people and what we see in different experimental conditions. So we asked, can we still use this technique to identify the cells in different experimental conditions? So to do this, we used a stimulus that is used in many different talks this weekend, a cholinergic stimulus, methacholine. So here we just looked at conventional staining at first. So here we have a mouse trachea. You can see the cells outlined with membrane tomato and magenta. And you see in green CCSP staining, or CC10, or SCGB1A1, or secretoglobin, your favorite name for this protein, that is a marker of the secretory cells. So this is at baseline. And then with methacholine stimulation, we see a lot less of the CC10 staining. And presumably, it's because the secretory cells secrete the secretoglobin. And so we're less able to identify the secretory cells using just conventional markers. So we asked, would the same thing happen with autofluorescence? So here's baseline, looking at the mouse trachea with autofluorescence. Here's FAD in yellow, NADH in blue. Now remember, the secretory cells are rich in NADH. So there are all these blue cells here that we're seeing. You see plenty of secretory cells, a lot of these blue cells. After methacholine stimulation, you still see these blue cells. They're still present. And we actually can quantify. There's some change in autofluorescence magnitude. We can still identify the secretory cells after methacholine stimulation. So in some ways, maybe this is better than conventional staining in certain situations. But one thing we noticed when we were looking at the cells after methacholine was that they looked a little different. And so I'm going to draw your attention here, and let's blow it up so we can take a better look at it. So here's the secretory cell, a little pixelated, but blown up at baseline. And here's after methacholine stimulation. And there's this like void within the cell. Now we're thinking, could this be the nucleus? Are we looking at a different level in the cell? So we did XZ optical cross sections here. So here's baseline, you see the nucleus. And then after methacholine stimulation, still you see the nucleus, but this void is still present. So what's in this? It doesn't have metabolic cofactors. So what could be inside the cell? What's in the void? So we hypothesized, could this be the secretory cell taking up the extracellular environment? Do secretory cells sample luminal contents? To get at that, we incubated with a cell-impermeable fitzy-dextran, and then stimulated with methacholine and looked to see what happened with the secretory cells. And this is what you see. So fitzy-dextran in green, and you can see that the secretory cells uptake the fitzy-dextran. And in fact, these are in membrane-enclosed vesicles. So secretory cells sample the luminal contents, luminal environment in membrane-enclosed vesicles. And this is interesting. Didn't know this would happen in the airway. Is there precedence for this happening in other tissues? In fact, in the intestine, there are goblet cell-associated antigen passages, or GAPs, where the goblet cells uptake luminal contents and present them to antigen-presenting cells. So here's a great example from this great paper looking at goblet cell-associated antigen passages, where you have dextran in red, nucleus in blue. That's in the intestine. And then looking in the airway, that's also a fixed. Here we have a live image. We can see dextran in green, nucleus in blue, and it looks pretty reminiscent to what we're seeing in the intestine. And here's the en face view. So we thought that, we called these secretory cell-associated antigen passages, or SAPs, because they resemble what we see in the intestine with intestinal gaps. Now, one part of the name of gaps is antigen passages. So an important mechanism, important physiological process that the gaps are involved in the intestine is delivering luminal antigen to immune cells basolaterally. So we asked, do secretory cells do the same thing here? So using a mouse that expressed mCherry under CD11C promoter, so all the APCs here are mCherry. We stimulated with methacholine in the presence of dextran to visualize SAPs, and then we saw that there was co-localization of these immune cells with the SAPs. This is even more evident here on the XC optical cross-section, looking at the APC. Here are the SAPs, and here we see co-localization here. Importantly, we don't show that the antigens are being passed on to the immune cells here, but at least there's some sort of co-localization going on. We next wanted to better visualize and quantify the formation of these SAPs. So we did high-speed imaging. Quite not the resolution we would have liked, but high-speed imaging to try to quantify this. And so I'm just going to let this play, and I'll walk you through what I suspect is happening. All right, so let me try to lay out what I think is happening here. So initially, what we have is uptake of luminal contents into these membrane-enclosed vesicles. This occurs and they move towards the lateral aspect, and you see that accumulation on the lateral aspect of the cell here, with additional uptake of luminal antigens on the luminal surface, up to a point at which we then see a massive secretion event of some of the luminal contents. Now, this model is very provocative and has a lot of more questions than answers at this point. For instance, we've seen both secretion and uptake by secretory cells. So how are these different processes regulated? Are they linked? So far, we've looked at secretory cells here, but secretory cells can become goblet cells. And goblet cells I've known to have different cargoes in different vesicles. How does that fit into all this? What are the roles of that? We're talking about CFTR here. What's the role of CFTR in this process? CFTR on the apical membrane, CFTR potentially on vesicles. How is this involved in both secretion and uptake? And also importantly, there's many implications for airway immunology. What antigens are taken up by the SAPs? Are they processed? And how are they presented to the APCs? So a lot of questions to look at. Fortunately, one of my colleagues in the Rajpal lab, Stephen Zwick, is working on this, but clearly a lot has to be done. So let me take a second just to summarize what we've shown so far. So we've developed this non-invasive approach to identify all 7 airway epithelial cell types that we hope to use in humans. Using this approach, we've identified secretory cell-associated antigen passages, or SAPs, which has important implications not only for airway epithelial function, but also for airway immunology and disease. Great. Lastly, I just want to thank everyone involved in this work, and particularly Jay Rajgopal, my mentor, for all his guidance and support, and the CFF for all their support as well. Great. Love to answer questions.
Thank you very much.
That's really neat.
Thanks.
So oxidation reduction is a sort of dynamic process, right? The balance between the two and mitochondrial.
Right.
So how much variation do you see when you do these different preparations? Because I can imagine taking out the trachea may trigger some shift in energetic status. And also the cells could vary in the amount of mitochondria. So how much variation is? And second of all, have you looked when you provoke them, say with the acetylcholine, which is an energy-dependent vent, I would assume, for secretion? Do you see a shift in the autofluorescence?
Yeah, fantastic questions. So the first one was, do the different preparations, what's the variability between that and the autofluorescence in particular? So there is some variability. The way we did this preparation, we actually didn't see as much as we'd have thought. We also looked across multiple days. And at least across 3 days, the NADH and FAD autofluorescence signatures stay relatively constant. So that was quite surprising, but that's what we saw. That was the first question. Second question, the perturbations. Very interesting point. So with methacholine, we're still able to see differences between secretory cells and ciliated cells, but it's interesting. The secretory cells have an increase in FAD, which one would think potentially could be happening if they need more energy, right, using oxidative phosphorylation. We haven't looked at other compounds. It'd be very interesting for cyclic AMP or other ways to stimulate the cells to look to see how these differentially change the autofluorescence between all the different cell types. Yeah, great question.
So I was just a little curious. Have you tried combining that autofluorescence kind of like imaging technique with like staining techniques where you don't need to use PFA to fix the cells?
Can you elaborate a little bit more? What do you mean?
So sometimes you can use some techniques where you don't need to actually fix the cells, so they're still live, so you can use some indicators. Have you tried that with the autofluorescence?
Yeah, great question. So you can use sometimes like live lectin staining to identify different cells and things like that. The issue is it'll interfere with the autofluorescence because a lot of especially on the 2-photon, there's a lot of, it's not as specific as we would like. And so there's like a lot of bleed through. But doing it sequentially could be something we could think about. Yeah. And so combining a lectin to identify a different cell could be another way to do it. Yeah, that could be a way to enhance the live imaging technique. Okay. Great idea.
My other question was, are you using like a single wavelength to kind of like do this, or can you like tweak the wavelength to kind of like contrast the different cells?
Yeah, great question. And you know, actually this work has been published in eLife, and we have all the methodology over there as well. The microscopy gets a little complicated in terms of the filters we need to be able to use to do autofluorescence, but we have tunable lasers, and so we're able to stimulate specific wavelengths of light right there. The emission's a little bit tricky when you're adding different fluorophores onto the autofluorescence. But yeah, great question.
Thank you.
Yeah.
Hey, so did you also try bright fields only? So only based on bright fields. I mean, maybe nuclei and cilia would be recognizable, but maybe the rest much less. But did you, or are you planning to?
No, that's a great question. We haven't looked at bright field. Yeah, that's something that would be really interesting to look into.
Okay.
Yeah, thanks for the suggestion.
Really beautiful data.
Thanks.
So the idea of the secretory cells actually surveying the environment. So do you have a model in which you might think in the case of CF, you have sort of the thick dehydrated layer may affect the ability to survey? Are there models that you can assess that in?
Yeah, great question. And I think the intestinal literature is really rich here for us. There's actually not as rich as we would like, but there's an important work done in terms of antigen sampling and immune tolerance in the intestine. How the mucus layer and how those changes are going to affect it, I don't know. It would be interesting to try that on here and induce a CF phenotype or induce a PCD phenotype, for instance, and see how that affects uptake. I don't know. That's a really interesting point that we should try.
I was missing the dynamic measurements of autofluorescence changes. When you put on the agonist,
yeah,
the function of time, see how fast, where's the peak, and what time, in what cells. There's information hidden in that. And I was also missing the purinergic stimulus to maybe see differences in the cell types.
Yeah, great points. It'd be great. We haven't done any purinergic stimulus. We only looked after 30 minutes of the methacholine stimulation, so I don't have a full time course over hours to characterize when it peaks and not. But yeah, those are interesting points that we should look at.
We have done this type of measurement in epithelial cells from the colon, and these changes in autofluorescence are very fast, actually.
Ah, interesting.
Very, very fast, because you trigger the calcium signal, which goes into the mitochondria, activates the mitochondrial respiration, and by that you change these fluorophores. They're very fast.
Right.
So maybe you can exploit that.
Yeah, great. Thanks. I'll look into it. Appreciate it.
Wenjie from University of Iowa. Great talk. Just curious whether you tried human tissues like human iOS.
Yeah, we've done some human tissues. We're still working on the kinks for the human epithelia. Humans are, of course, a little bit more challenging, a lot thicker epithelia, particularly if we're going to do it in live tissue explants. Potentially a lot more variability, like we were talking about as well, because there are various disease states. And even in normals, you can probably think there'd be a lot of variability there. But yeah, it's on the to-do list, a long to-do list.
Okay. Go ahead, last question.
Very inspiring idea. So my question is, where do the transitionary cells fit in your map?
Yeah, great question. So I think this goes back to what do we call cell identity? What's a cell state? What's a new cell type? And I think you ask one person, you ask like 10 people, they'll probably have 10 different answers in terms of that. So how these changes over time will be really interesting. So looking over, of course, days with potentially like IL-13 or other types of stimulus to see how cells transition, secretory cells to goblet cells. It'll be really interesting to see how these autofluorescent markers change.
So are the transitional cells included in your map or they are excluded?
Yeah, great question. So we only use those conventional markers, KRT5, CC10, acetylate tubulin, and the rare cell markers. So we didn't look at any combinations of different markers to look to see transitioning populations in this. But that's a great point.
Thank you again.
Thank you so much. Appreciate it.
All right. And I just want to wrap up by saying thank you very much to the audience for being so engaged and providing excellent questions. We hope you've enjoyed discussing the alternative mechanisms of CFTR, gating and potentiation, and everything else today. So hope you enjoy the rest of the sessions.